Okay. Sorry. Sorry about the change in background too. On the other side of the wall right here is where Nathan's asleep, so he's just getting over strep, and everybody else in my house is kind of blah. So that's why I had to wait till so late to do this. But also, instead of having to run from my kitchen to the bedroom, I am now just a wall away. So, okay. Last thing was Chronobacter, the necrotizing enterocolitis in infants, that's awful. It is associated with contaminated infant formulas, and that just makes my mama heart hurt. So anyway, okay, that is it for the gamma proteobacteria. Now we're on to the delta proteobacteria, which is slide 39. So let's see. They are SRBs, or sulfate reducing bacteria. So the way that they reduce the sulfate is they use that as the final electron acceptor in their electron transport chain. They can be predators of other bacteria, and they can be pathogenic. So the types are on slide 39. We are going through the Greek alphabet. Epsilon proteobacteria is on 40, slide 40. It is gram-negative, and it is microaerophilic, so it doesn't need a lot of oxygen. We talked about that. And here you have Campylobacter, which is gram-negative, and they are spiral-shaped rods. They're aerobic because it's an epsilon proteobacter, so it's microaerophilic, but it's still aerobic because it still uses oxygen, even though it doesn't need a lot of it. Often found in chickens, but it can affect humans if you don't cook your chicken enough. Then you've got Helicobacter, so gram-negative spiral-shaped rods. They're aerobic, but also they're the microaerophilic, so micro, needle, aerophilic, lacks a little air. Can damage the inner lining of the stomach. And the Helicobacter can cause chronic gastritis, peptic ulcers, sometimes stomach cancer, so no bueno. Next, we have non-proteogram negative bacteria, so that slides 43 and then 48 through 50. You've got the phyla cyanobacteria. So these are your bacteria that are oxygen photosynthetic bacteria. They're nitrogen fixing. But remember we talked about this last unit where these are photosynthetic bacteria. Prochlorococcus, So they're the smallest photosynthesizer and one of the most abundant on Earth. Next, you have the phyla Chlorobi and Chloroflexi. So they are anoxygenic photosynthetic bacteria. This is your green sulfur bacteria. So you've got, I wish I knew how to write stuff on these videos. I'm sure you can, but if y'all watch this before class tomorrow and have any questions, I'll try my best to bring my folder and I'll answer them. Coughing child, hold on. Okay, so anyway, for the cyanobacteria, it does have a photosynthetic formula. So you've got 6 CO2 plus 12 H2O plus light energy, and that's going to yield your C6H12O6 glucose plus 6 O2, so your 6 oxygens, and then 6 H2O, so 6 waters. That's your prototypical photosynthetic formula. But for the phyllochlorobene, chloroflexi, those anoxygenic photosynthetic bacteria. You've got the CO2 plus 2H2A, and I'll get into the A in a second, plus light yields CH2O, which is a carbohydrate, plus 2A plus water. So the A is a placeholder. So it can be, sorry y'all, my nose itches. It can be any electron donor. So some of the most common are water, H2S, H2, any other electron donor can take the place of that A. Next we have Cytophagia, Fusobacterium, and bacteroids. Cytophagia is aquatic. Fusobacterium is in our mouths. And the bacteroids are the largest. But there is more information on those on slide 48. Next, in this non-proteo gram-negative bacteria, you've got spirochetes. So like springs. They are motile with axial filaments. So within the spirochetes, you have Treponema pallidum. You have syphilis. You don't have syphilis. You know what I'm saying. Sores. You got sores where there shouldn't be sores. Just leave it at that. And it is an STD. It is sexually transmitted. Next, you have Borrelia burgdorferi. Borrelia burgdorferi, Lyme disease, and that's ticks and lice. And that causes the bullseye rash, fever, fatigue. And then you've got the Borrelia recurrens, which is relapsing fever. So recurrens, recurring, relapsing fever. Let's see. Next we have Deinococcus radiodurans, Conan the bacterium. This bacteria can survive the harshest conditions on Earth. It can survive 50,000 grays, which is a unit of radiation, which is 1,500 times what it would take to kill a human. It has this like rapid repair of radiation damage. It's insane. That's why it's called Conan the bacterium. You can't kill it. Let's see. Next we have Thermus aquaticus. So it is heat stable. And its enzyme is the TAQ polymerase. So it is extricated and used in the polymerase chain reaction, so PCR. So when you do PCR testing, it uses that TAQ polymerase. Next, we have the gram-positive bacteria. So Firmicutes on 52, 54, and 55. So let's talk back. Let's talk about DNA for a second. We know that when that backbone is made, that those base pairs, we know that A is always with T and C is always with G, correct? So adenine always pairs with thymine and cytosine always pairs with guanine. So in these gram-positive bacteria, or two of them, you're going to have low CGs. You're going to have low cytosine and guanine. So the Firmicutes, there are a lot of examples, but they have low guanine and cytosines, and they include endospore forming bacteria. So you've got bacillials, clostridials, staphylococcus, lactobacillus, streptococcus, enterococcus, listeria, and monocytogens. So like there's a lot. Next we have the tenericutes. They're mycoplasms, and they also have the low guanine and cytosines, and that includes the mycoplasms which have high pleomorphic, or a high number like of pleomorphic. It lacks cell walls. Pleomorphic, lacks cell walls. So mycoplasm pneumoniae. There's a pneumonia that's there. Yes, honey. You're hungry. Okay. Well, I'm letting him eat. He hadn't eaten since like Friday or Saturday. He's had strep. Yeah, buddy. Next, in this gram-positive bacteria, I'm gonna get to see my other child this time. Just so you know, he got on underwear. He's been asleep. What would you like to eat, bud?
Cereal.
Cereal with yes milk or no milk?
No milk.
Okay. I'm not gonna stop the video, so just hold on 2 shakes, okay? Get to look at the back of my couch. We can't turn the TV on, baby. Because Mama's making a video for school. Here, I'll wrap you up. And you want on my phone.
Or play a game on your phone?
Yeah, you can play it on my phone.
Hello, what's your play game? I want to...
Yeah, he just gonna get comfortable. He's hungry.
Just write him up and you were sitting around.
It's okay. I'm doing my video right now.
I'll put it right here so I can
It's
eat it
facing the
later.
couch right now.
I'll eat it later.
You told me you were hungry.
I will eat some.
Oh,
They're all like little. Little. And I'll eat it later. They're having the phone, Dad. So wait a minute.
You okay?
You're punchy.
Like Daddy. That is into his yet.
Okay, you get your snap and then you go back to bed, okay?
Here.
Mom.
Y'all got to see my absolutely destroyed living room. That's what happens when you have a 5-year-old and a 3-year-old. Y'all, sorry. I just didn't feel like stopping the video and uploading more than 2 videos. Okay. Now we're on to actinobacteria. So these actually have a high number of cytosine and guanine. They're highly pleomorphic. And so that's the plasmas as well. They're highly pleomorphic, so they don't have cell walls. Actinobacteria, they have the high guanine and cytosine counts. They're highly pleomorphic, and they can live in soil or aquatic environments. They're mostly aerobic. Their distinctive feature, presence of several different peptidoglycans in the cell wall. That's what makes them distinct. Okay, so next we have in our Staphylococcus. I have all these written out, but I want y'all to look them up. Oh, for the actinobacteria, you've got Mycobacterium on slide 60. You've got Corynebacterium on 61. And then on slide 62, you have Bifidobacterium, Gardnerella, Propionibacterium, Frankia, and Streptomyces. All those are on 62. And there's more information on there. All right. So that is it for the prokaryotic classification one. Next, we're on to the eukaryotes and fungi, the mycology slideshow. So, okay, the functions or features of fungi. Their cell walls are made of chitin. They can be unicellular or multicellular, heterotrophic, which means they absorb the food after digesting it out of the body. They're heterotrophic. They reproduce asexually or sexually. They produce spores. Heterotrophic. Chemoheterotrophs. So they absorb the nutrients from a substrate. So like wood, tissue, cells, organic matter, that's where they're getting their nutrients. They adapt to places that are harmful for bacteria. That's why they can thrive there. They grow better at a pH of about 5, so more on the acidic side. Almost all molds are aerobic. Almost all yeasts, both of these are fungi. Almost all yeast are facultative anaerobes. They are more resistant to osmotic pressure than bacteria, so they can grow in high sugar and high salt concentrations. They grow on substances with low moisture content and needs less nitrogen than bacteria does to have the same growth. They can be capable of metabolizing carbohydrates such as lignin, like in wood, that bacteria can't. So they can break down these carbohydrates and thrive in places where bacteria can't thrive. And all of this enables fungi to be able to grow in some really strange places. So like athlete's foot, ringworm, all that fun stuff. Yeah, all those fungi. Not a fun guy. All right. Next we have lichens. And so what lichens are is a combination of a fungi and an algae together. An algae or a cyanobacteria. And they are, it is a mutualistic relationship. So they are both benefiting here. The fungi is typically the dominant partner of the partnership. The algae is usually green algae or the green-blue cyanobacteria. You okay?
Yeah.
Good. So with this, we'll get into how they benefit here in just a few minutes because I wrote it on the next page. Yeah, right on the next page. Mycorrhizae is the next one. Mycorrhizae. It is a mutual relationship between plants and fungus. So, there are two types. There is the ectomycorrhizae, so it's formed by Basidiomycota and some Ascomycetes Occur mostly in woody plants of the temperate zone. So this is the less common type. The Ectomycorrhizae, it's the less common type. So it has what is called a harding net. So it has hyphae that grow inward and extend into the root of the plant. And this extension of that inward hyphae is where the nutrient exchange between plant and fungi happens, or fungus. The next one is the arbuscular. So it's formed by glomerulomycetes, and it occurs in 80% of plants. So it's the most common. The arbuscles, they are highly branched hyphal structure that provide the nutrient exchange between the fungus and the plant. So in a lichen, the benefit is that the algae provides the fungus with a food source by photosynthesizing and growing. And the way that it's able to grow is the water and nutrients that get absorbed by that fungus that's growing on it. So the algae grows because there's moisture that the fungus brings in, and then the fungus grows because the algae is providing a food source as it photosynthesizes and grows, and it just is a nice little circle. In the mycorrhizal, the fungi benefits from getting carbohydrates from the plant that it photosynthesizes. It's the carbohydrate that is the result of photosynthesis. And the plant benefits from having an increased water and nutrient supply from the fungal absorption. So next you have definitions. So I'm going to tell you what slide to go to. To define these, but I'm not going to define them for you. On slide number 4, you have hyphae, mycelium, thallus. I'm trying to make sure there's not another 4. On slide 5, you have septate, pseudohypha, septate hypha, and then the coenocytic hyphae. On 6, you have yeast. On 8, you have mold and mildew. On 9, you have mushrooms. And on 11, you have pretty much the rest. Plasmology, karyogamy, and then an ascus is an internal sac that holds the sexual spores of one of the I forgot to write it down. We'll get to it. But an ascus is a sac inside that holds sexual spores. Alright. Name and describe 2 types of asexual spores. Kinetiospores, which are spores that are not in a sac or encased. And then you have the sporangiospores, which are formed in a sporangium. So examples of fungi with sporangiospores: Penicillium, which is Canidia. Yeah, I'm sorry, I can't read my handwriting. Coccidioides, emets. Blastoconidia and Chlamydia conidia. It's the one that we looked at in class that day. There's 4 examples of those asexual spores. And then, let's see. Another example of asexual spores. Not asexual spores, I'm sorry. Hold on, time out. I read all that wrong. All of those examples I just gave are examples of conidiospores. So, I know I said it was for sporangiospores, but I wrote it backwards in my notes. So, what I just gave you were examples of conidiospores. The next examples I'm going to give you are examples of sporangiospores. It's really easy. It's rhizopus, which is bread mold. Sporangiospores, rhizopus, bread mold. Conidiospores, penicillium. There's an example. Call it done. Name and describe 3 types of fungal sexual spores. Ascomycota, which are sex spores, are born internally. Ascomycota, that's where your ascus is. Sex spores are born internally in a sac called the ascus. It can be asexual, but mostly asexual. Basidiomycota, which are sex spores born externally in a club shape called a basidium. And then the third type is Zygomycota, and these are sex spores with thick cell walled resting spores called zygospores. Let's see here. Fungal classification. Sac fungi are the Ascomycota. Club fungi are the Basidiomycota. Zygote fungi are the Zygomycota. Arbuscular fungi are the glomerular. Why is this so hard for me? Glomerulomycota. There we go. The chytrids are the Chytridiomycota. Pretty straightforward. On slides 17 and 18 for the dimorphic fungi, they have the ability to switch between morphologies in a life cycle, so like between yeast and mold. You okay?
Yep, I'm fine.
Okay. They start in soil, and when they're inhaled or inoculated by injury in mammals, it undergoes a process to become pathogenic yeast that can cause health issues. So that's why it's important in the medical field, is because if they get into your bloodstream and you are affected, it's going to go through that morphology and turn into something that is potentially problematic. Slides 21 and 22 about Zygomycota. They're bread molds. They're well developed, multinucleate form. Coenocytic. They're coenocytic. So coenocytic. They have a multinucleate form, which is formed from multiple nuclear divisions. They're haploid mycelium. They're weak pathogens. An example is the rhizopus, so bread mold. Slides 24 and 27, the Ascomycota. They're the sac fungi. They're the largest group. Most are plant pathogens. Some are beneficial. Truffles, black truffles, watch cooking shows. Morchella, cup fungi, the little puff balls. What you see in your yard. Slides 29 and 31, the Basidiomycota, those are your club fungi. They create fruiting bodies, mushrooms. They can be a food source. They can be medically important. The puffballs and what you see in your yard. Those are the Basidiomycota. Not the Ascomycota, I'm sorry. My bad. It's late. With the Ascomycota, I would just go with truffles. The Basidiomycota, they're ectomycorrhizal symbionts. So they make an underground resource sharing network. The Wood Wide Web, I think, is what it was referenced as in your. They're important to decomposition, so they're important decomposers. They are a food source, but they can also be destructive. And also they can be medically important, so see slide 29 for that. Mushrooms, toadstools, puffballs, rusts, and smuts, those are the bad ones. So Chytridiomycota, the chytrids, this is on slide 35. They're the earliest lineage of fungi, and they are the only ones with flagellated spores, so the flagella on the spores. They're single-celled with branching hyphae and rhizoids, and they're found in fresh water, soil, and mud and rumen. Can be pathogenic. Saprobes are parasitic. Yeah, that's it for that one. Next is the Glomeromycota, or the arbuscular fungi. So this is on slide 20, where it's 80% of all higher plant species interact with a mycorrhizal partnership with these. So yeah, that's the Glomeromycota. Okay, that's it for that slideshow. Blah. Next, y'all, I'm sorry I keep moving y'all around. I love you too, baby. Next, we have the eukaryotic microbes and protists slideshow. So again, I'm not going to define these words, but I'll tell you the slide number. Let's see. On slide number 1, you can find protists, protozoa, algae, planktons, zooplankton, and phytoplankton. On slide 4, you'll find seaweed and another reference to phytoplankton. 12, the undulating membrane. 16, the pellicle and the cyst. And 24, the rope trees, macronemes, and the apical complex. All right, name 3 groups of algae. All right. Name 3 groups of algae that produce toxins. So algae that produce toxins. Diatoms, dinoflagellates, and red algae. All right. Diatoms, dinoflagellates, red algae produce toxins. I don't think red algae produces as much as the other 2. Name the algae that is used to make sushi wrap. Nori. It sounds like it belongs on sushi. And source of algae and carrageenan, the gelling agents. So the carrageenan is a gelling agent for vegans that is not taken from animal parts. Gelatin is the protein from animal parts that is used for the gelling agents. And then the agar is the gelling agent that we use for growth. Let's see. Name the multicellular algae that includes seaweed, such as kelps, brown algae. Brown. Name 3 algae that can cause algal blooms. Algal blooms no good. Cyanobacteria, which is the blue-green algae. Dinoflagellates, which is microalgae or red tide. And diatoms, which is the microalgae or the red tide. So the 2 algae that causes red tide, dinoflagellates and diatoms. How do algal blooms harm people and other organisms? They can produce toxins. They can be too dense to get through for the organisms that live in that environment. They can use up all the oxygen in the water, and they can release harmful gases. So it's just not good all the way around. Okay, true or false? Paralytic shellfish poison, PSP, can be destroyed by cooking or freezing. No, it cannot. It cannot. It is usually caused by those algal blooms. So if there's shellfish in that area, you cannot eat it. Let's see. Know the following protozoans. So all that's listed here. You need to know how they're transmitted to humans or how humans are affected by the protozoan, the diseases they cause, and if there are any vectors. So there are 4 ways typically for a protozoan to infect a human. Fecal, oral, contaminated, food or water, person to person. So that contaminated food or water, it's also fecal, oral. Person to person, vector borne, or predator prey transmission. Most of what we're going to be looking at is not predator prey. So Giardia intestinalis, it is transmitted the fecal oral route, so it's excreted as a cyst in the feces of the affected and then ingested. As far as disease and vectors, I didn't see any. But let me pull it back up real fast. Good job. Get it out, buddy.
Can you get this rock? Oh. It's a rock.
It's a rock. Can you bring it to me? You want to come say hey in my video?
No.
Yes.
I'll see myself.
Yeah, you can see yourself. Come on.
Turn it around.
No, you gotta walk over here. If you want me to fix the video, you gotta walk over here.
Okay.
Okay.
Can I take my covers off?
Yes.
Unless I'm cold again.
Unless you get cold.
Back to
So, it doesn't say any diseases or any symptoms of diseases.
Hey!
Say hey.
Doing it.
Nathan.
Fix it.
Fix it. Excuse me.
Please.
Thank you.
What's a thank you?
Here.
Is it a word?
It is. Go sit down back under your covers and eat your cereal. Sick boy. Okay. So it's unicellular with flagella, so it's a parasite without mitochondria. Found in intestines of humans and other mammals, and it is excreted in feces as a cyst, so it survives in environments before being ingested by another host. It doesn't tell me any symptoms, any diseases, or any vectors, so that's all I got. Okay, next we have Trichomonas vaginalis. So it is also unicellular with flagella, parasite without mitochondria. It has an undulating membrane, so it consists of a membrane bordered by a flagellum. So it does not have a cyst stage, so it must be transferred from host to host quickly before the desiccation occurs. So it's either sexual contact or toilet or a towel. And it's got to be pretty immediate or it's going to break down.
That's a PP word.
It is. Next we have Trypanosoma brucei. And that is the African sleeping sickness. That one's not good. It is transmitted by the bite of a tsetse fly. And it has different, has 2 different, like it can be acute or it can be chronic. And that is on slide number 21. Sorry, I have to go between pages here. There you go, buddy. You get fever, skin lesions, rash, swelling, swollen lymph nodes on the back of your neck. After a lot of weeks, it can become meningoencephalitis, so infection of the brain and the fluid that surrounds the spinal cord. So yeah, it's very, very not good, especially given that it occurs in places where medical treatment is not always the best. So yeah, no bueno. Next we have Trypanosoma cruzi, and that causes Chagas disease. The kissing bug bites your face. It's a bug that bites your face and makes you sick.
I don't love kissing bugs.
Yeah, good. You shouldn't.
They hurt.
Yeah, they probably do. Next, we have entamoebic histolytica. And it's a fecal oral. It's you ingest the ingestion of a cyst from infected feces. Yeah, it's kind of gross. And that's what causes amoebic dysentery that we talked about in the previous video. Next, we have Toxoplasma gondii. So, eating undercooked or contaminated meat or exposure to infected cat feces. Or a mother-child transmission through pregnancy. And it's toxoplasmosis is what it causes. And that, let's see here. Get it out, buddy. Is on, if you want more information, it's on slide 24. Next is Plasmodium falciform. Yeah. Plasmodium falciform. Falciform. Infected mosquitoes carry the disease from one human to another, acting as a vector. So the mosquito is technically the vector here. So infected humans transmit the parasite to the mosquito, whereas the mosquito doesn't get affected at all by the malaria that this causes, but it's transmitted via mosquito bite. And so the slide 25 has like the whole life cycle, but we're going to go through that here because the next question is define the following components of the Plasmodium life cycle. So the hypnozoite is the dominant stage. So it just kind of chills in the liver and can flare up anytime it wants, basically. So the sporozoite is motile. So it's the spore-like stage. And this is what infects and goes into the liver. And so the sporozoite chills in the liver and then matures into the schizozoite. So once this stage occurs, once it matures to this point, it starts to rupture. And it ruptures and releases the merozoite. So this is the blood stage. So it breaks out of the liver and enters the bloodstream. So remember, this is all about malaria. Malaria caused by infected mosquitoes. Hate them. So we're at the merozoite, so the blood stage. And so it breaks out of the liver and enters the bloodstream. And it invades your red blood cells. And it grows and divides and destroys your red blood cells. So once it gets in your blood, you've got the gametocyte. Fun stuff. So it's male and female, and they're ingested by the mosquito when it has a blood meal. So this is where the mosquito then becomes the vector. So the mosquito ingests the gametocytes, which is the male and female. And this is when this sporogonic cycle happens. And then it produces the ookinete, which is motile, and it's an elongated zygote that develops into the oocysts. So the cysts then develop and rupture, and it releases the sporozygotes, and those sporozygotes go to the mosquito's salivary glands. And once it gets to the salivary glands, it's the trophozoite, and that's the feeding stage. So then it goes into the salivary glands. The mosquito has a blood meal, and it's in those salivary glands, and it gets injected into the human to infect another human. So it absorbs the nutrients and starts the whole process over. Once these are released into the bloodstream, and it goes into the liver, and it just starts the whole thing over again. That makes sense, I hope. This is all on slide 26. Okay. Next, we're on the eukaryote parasites slideshow. I feel like this one's the shortest. The least number of pages I have. All right. Features of helminths. Okay. Helminths are worms that live in or on humans or animals, and they cause diseases and malnutrition. They're eukaryotic. They're in the animal kingdom. Multicellular with no GI tract, no intestinal tract, and a reduced nervous system. Locomotion is either reduced or complex. It has a very complex lifestyle, and it can be dioecious, which is male in one of the worms and female in the other. It can be monoecious or hermaphrodite, which is both male and female in one worm. Let's see. Helminths that are parasitic to humans are the platyhelminthes, the spiny or horny-headed worms, and the nematodes or roundworms. For the helminths listed below, make sure you know, again, the same stuff that we needed to know earlier, how they're transmitted, their host before they're transmitted to humans, and what diseases they cause. Now, I'm going to get to those in just a second. I'm going to answer the arthropod vectors question, and then I'm going to show you my little chart that I made, and hopefully I'll be able to get that into something I can print off for email or something, so that kind of puts all that in one spot. Okay, so arthropod vectors are exactly as they sound. They're animals that are arthropods that carry pathogenic microbes and that can infect humans. So like spiders, mites, and ticks in the arachnoids. Four-legged, you got the crayfish and the crabs. Insects, you got bees, flies, mosquitoes. I mean, like, any animal that's an arthropod that can carry a disease to a human is going to be an arthropod vector. Okay, so I made this nice little chart. I don't know if it's... My handwriting is atrocious, I'm aware. So, you good? So the Chinese or Oriental liver fluke. It is transmitted by eating raw or undercooked fish or crabs or crayfish. So the hosts before it gets to humans are the fish, the crab, or the crayfish. And it's severe symptoms and illness. That's all the slide says. It can easily be prevented by freezing and cooking the animals properly. Same goes for the...
J-E-E-P.
What?
J-E-E-P.
What's that spell?
Jeep.
Jeep. Good job. He's reading now. And he reads everything. So proud of him. Next, you have the Opis.
T-R-U-C-K.
What?
T-R-U-C-K.
Is that a truck?
Yep, that's truck. Spell. Circle.
The Opisthochus, which is the other like random flatworm. It is also transmitted by eating raw or undercooked fish, crab, or crayfish. It is also found in fish, crab, crayfish. And it causes indigestion, abdominal pain, diarrhea, or constipation. And again, it can be easily prevented by preparing, so cooking and storing the fish properly. Fasciola hepatica, that's next. That's the common liver fluke or sheep liver fluke. So you get this by eating raw watercress or undercooked watercress or other contaminated water plants that have been infected with the immature parasitic larvae from the Fasciola hepatica. So the host before us is sheep or cattle, and it affects your liver and your bile duct. Causes diseases in those areas. Okay, next we have the tapeworms. So we have the Tinea saginata, which is the beef tapeworm. So in all of these, you're eating undercooked beef or pork is how you get this. And it gives you a tapeworm. So you have terrible abdominal pain, no appetite, diarrhea, weight loss. Weight loss is the big side effect from a tapeworm. The Tinea solium, that is the pork tapeworm. And then the Tinea asiatica, it's the Asian tapeworm. So its host, the Asian tapeworm's host before us is pigs. So it presents more like the beef tapeworm, but it's found in pork. The visible signs of the Tenacias, the most visible sign is the passing of the proglottids. So when you poop out segments of a tapeworm. The abdominal pain, no appetite, weight loss, and upset stomach, that just goes with it. The function of the scolex in the tapeworm is the anterior part, so it's got the hooks and the suckers, and that's what attaches it to you, and it just, when you eat, it eats, and it just gets longer and longer and longer and longer. Those proglottids just keep adding up on the end, and then you're pooping them out. But they're still making more. That's how they get going. Because in each proglottid is a fully mature sexual reproductive system. That's the next question. What is a proglottid? So it is the segments of the tapeworm, the mature segments of the tapeworm. So the further you get from the scolex, the more mature it is because it's been on there longer. It contains a complete sexually mature reproductive system, and it must be removed or it will reproduce either inside the host, like it's going to keep going. So you have to get each piece out. What are cysterioses? They're caused by the pork. It's caused by the pork tapeworm, so the Tinea solium, and it is a disease that causes seizures. 2 tapeworm adaptations when in the human digestive system. It has a skin-like covering that protects it from digestive juices, but it's also porous enough to let in food. A tractor detector. That's cool. Okay, so next we have the spiny or horny-headed worms. They are very rare in humans. If you ingest an infected host, then you're going to get it. And the hosts are typically rats, raccoons, and pigs. And it causes digestive issues that go along with abdominal pain. Last but not least, we have roundworms, or the nematodes. So the filarial nematodes are what I'm gonna go through that too. So the roundworms, it's ingestion of contaminated water, but the filarial one is a mosquito bite. That's how you get it. And the host before us is mosquitoes. And the filarial roundworms cause lymphatic filariasis. And so an estimated 90% of LF is caused by the, hold on, I want to get the word right, as right as I can. Sorry. The Wuchereria bancrofti. I knew it was going to be weird. That's why I abbreviated it. The bancrofti is caused by the W. bancrofti. So 90% is the Wuchereria bancrofti. The LF. Mosquitoes can carry, which mosquito can carry both lymphatic, the LF that is caused by the Wuchereria bancrofti and the Brugia species. So, which can carry both? See, I saw 2 on there that carried each one. I got the Aedes and the Mansonia. So if she told you something specific in lecture, go with that. Chronic LF is called elephantiasis. And I hate that.
Don't say that word.
I'm sorry. I don't like that it's called that because it has a bad connotation, but it's still right. Let's see. Next we've got pinworms, the 3 ways to diagnose and treat. Just buckle up. All right. You can inspect. So you can look for worms in the perianal region 2 to 3 hours after the person you think that has it goes to sleep. You can do the tape method. So you use clear tape first thing in the morning to collect what you think might be eggs from around the anus first thing in the morning. Or you can scrape under the fingernails and look at that under a microscope, because this is based on the thinking that people scratch what itches. Just throwing that out there. Is he doing it so fast? You've got both over the counter and prescription meds. Reinfection can occur, but it is totally preventable. Okay. Next we have 3 soil transmitted plants. Ascaris, so slide 15. Hookworms, slide 21 and 22. And whipworm, slide 16. Zoonic roundworms, 2 examples. Toxocara canis and Toxocara cati. One is from a dog, one is from a cat. 3 examples of zoonic hookworms. There are 4. Anclostomia brasiliense, A. caninium, and then the A. calinicium, or the Uncinnaria stenocephala. Why are words hard? Why do I make them so hard? yeah, so there's that. And then what causes and how are they different from each other? Cutaneous larval migrants.
Doors.
What?
These are keys for doors, and his keys belongs to the transmitters.
We have cutaneous larval migrans, which is a skin condition caused by the animal hookworm. Oh, there's larval infection in humans, and it's red and it's itchy, but it's just in the skin. The cutaneous, the CLM is in the skin.
What are you talking about?
I'm talking about microbiology to my students. Visceral larva migrans. It's where the larva, the Toxocara larva from the two zoonic roundworms, where that larva starts to infect the organs. So it's typically the liver or the central nervous system. And you've got fever, fatigue, coughing, wheezing, abdominal pain, all of that fun stuff. Then you've got the ocular larval migrans. So the OLM. It's just like it sounds, ocular. So it infects the eye. That larva infects the eye. Causes vision loss, inflammation of the eye, retinal damage. It's no fun. And it's all caused by the Toxocara larva infecting the body of a human. Is that it? Is that all of it? That's all of it, guys. Yay! So, okay, that's it. I'm going to work on getting this chart into a computer at some point, probably tomorrow. Right now, my goal is to get my kid back to sleep. Do you want to come say bye?
Can you turn around?
No, you got to come over here. The living room's a mess from where you and Sissy were playing earlier. And I didn't have the energy to pick it up.
Say bye to me.
Welcome to motherhood. You want to come say bye?
Can I say bye to me?
You can say bye.
Bye, me.
Hey, tell them good luck on their test.
Good luck.
Blow them a kiss. And tell them, do you feel better?
Everybody.
All right, guys. I will see y'all tomorrow. Bye.